General Biology 1, Module 4, Cell Cycle, Meiosis. Most essential learning competencies are characterize the phases of the cell cycle and their control points, describe the stages of meiosis given the diploid cell where 2n is equals to 6, discuss crossing over and recombination in meiosis, explain the significance or applications of meiosis, and identify disorders and diseases that result from the malfunction of the cell during the cell cycle. For learning objectives, you're going to describe meiosis, identify and describe the phases and stages of meiosis, explain the crossing over and recombination in meiosis, and understand the significance or applications of meiosis in living organisms.
Identify disorders and diseases that result from the malfunctions of the cell during the cell cycle.
Before we proceed to our lesson, let us have a short video about the topic.
The journey of life starts with a fascinating race. Starting with millions of sperm, only one, the fastest and the strongest, will reach the finish line. Some fall out of the race, getting lost in the rush through the narrow fallopian tubes, while some are stopped by natural obstacles which protect the woman's body from intruders. Those who make it closer to the end compete fiercely with each other. Together, they attack the egg cell. Their only goal is to be the first one to enter the egg. Only one will make it through, but the others simplify this mission by slowly dissolving the membrane of the egg. Finally, we have a winner. The winning sperm enters the egg and conception occurs. Half the man's and half the woman's chromosomes combine. This creates a unique DNA code which is needed to create new life. These combined chromosomes form the first cell of the new baby. This is now called the zygote. The zygote starts to divide into a cluster of cells and begins to travel to the friendly and safe habitat of the uterus. When the zygote is divided into a bubble of cells, a blastocyst is formed with an inner layer of cells making up the embryo and an outer layer which will protect and nourish the embryo. The blastocyst then finds a comfortable place to stay in the uterus for the next 40 weeks to develop and transform into a human being.
From the given video, sexual reproduction relies on a special type of cell division called meiosis. One of the provoking questions is, why do you share some but not all characters of each parent? Let us recall, class, what is DNA? DNA is short for deoxyribonucleic acid, and you probably heard that it's what makes you unique. And based from the given picture, it is a long chain of molecules that looks like a twisted ladder, and the rails are made of sugar and phosphate molecules. And if you still can recall during your junior high school, each rung is a combination of 4 different nucleotide bases. They are adenine, guanine, cytosine, and thymine. And the strands of DNA contain genes and chromosomes. Genes are like genetic paragraphs written using nucleotides, and chromosomes are like genetic books that contain large numbers of genes. And as we all know, the human body has 23 different chromosomes. Each cell has 2 copies of each chromosome, one from your mother and the other from your father. This makes a total of 46 chromosomes. Gametes are formed through meiosis, and this is also known as reproductive cells that unite during sexual reproduction to form a new cell called a zygote. And there are two processes of formation of male and female gametes. We have the spermatogenesis and the oogenesis. When we say spermatogenesis, this leads to the formation of sperm cells, whereas oogenesis, it helps in the formations of egg cells. Based on the given illustrations of the spermatogenesis, the primary spermatocytes contain a diploid complement of chromosomes, which is 46 chromosomes or 2n, where n is the number of haploid chromosomes. The primary spermatocyte then undergoes meiosis I to produce two haploid secondary spermatocytes. The haploid secondary spermatocytes undergo meiosis II to produce four haploid spermatids. Each spermatid begins to grow. Thus, this develops into mature spermatozoa, also known as sperm cells. The primary spermatocyte gives rise to two cells. The secondary spermatocytes, which is in turn produce four spermatozoa. In oogenesis, the cell starting meiosis is called primary oocyte. This cell will begin the first meiotic division but be arrested in its progress in the first prophase stage. At the time of birth, all future eggs are in the prophase stage. This results in the primary oocyte finishing the first meiotic division. The cell divides unequally, with most of the cellular material and organelles going to one cell, called a secondary oocyte, and only one set of chromosomes and a small amount of cytoplasm going to the other cell. This second cell is called a polar body and usually dies. A secondary meiotic arrest occurs, this time at the metaphase II stage. At ovulation, this secondary oocyte will be released and travel toward the uterus through the oviduct. If the secondary oocyte is fertilized, the cell continues to the meiosis II, completing meiosis, producing a second polar body and a fertilized egg containing all 46 chromosomes of a human being. Half of them, of course, coming from the sperm. What determines the gender of a baby? A child's gender, either a male or a female, is determined by the chromosome that the male parent contributes. Females have XX chromosomes and males have X and Y chromosomes. So men determine the gender of a baby depending on whether their sperm is carrying an X or Y chromosome. An X chromosome combines with the mother's X chromosome to make a baby girl XX chromosome, and a Y chromosome will combine with the mother's X chromosome to make a boy having an X and Y chromosome. Alright, the key difference between mitosis and meiosis is the way the chromosomes uniquely pair and align. Metaphase is a stage during the process of cell division, mitosis or meiosis. Usually, individual chromosomes can be observed in the cell nucleus. However, during metaphase of mitosis or meiosis, the chromosomes condense and become distinguishable as they align in the center of the dividing cell. Metaphase chromosomes are used during karyotyping procedure that is used to look for chromosomal abnormalities. What is meiosis? Meiosis is the process by which sexually reproducing organisms make their sex cells, sperm, and eggs. During meiosis, a specialized cell called a germ cell, which is any biological cell that gives rise to the units of an organism that reproduces sexually, which splits to make 4 new sex cells, each with half the number of chromosomes as the original germ cell. Each of the 4 sex cells has a unique combinations of DNA, meaning no 2 sperm or eggs are genetically identical. Meiosis is a 2-step process that occurs in the reproductive organs. And the process is divided into 2 types. Meiosis 1 reduces the chromosome number to half, and it is known as reductional division. Meiosis 2 is just like the mitotic division. Meiosis 1 consists of the following steps. We have the interphase, the prophase 1, the metaphase 1, and the anaphase 1. Just like the mitosis, meiosis also consists of preparatory phase, the interphase. The nuclear envelope remains intact, and the chromosomes occur in the form of diffuse, long, fold, and indistinctly visible chromatin fibers. In animal cells, a daughter pair of centrioles originates near or amidst already existing centriole, and thus an interphase cell has 2 pairs of centrioles. Prophase 1 is the longest stage of the meiotic division. It includes the following substages. We have the leucotene, zygote, tachytene, diplotin, and reakinesis. In the leucotene stage, the chromosomes become even more uncoiled and resemble a long thread-like shape, and they develop reed-like structures called chromomeres. In zygote, synapses happens. There's a pairing of homologous chromosomes. In tachytene, there's exchange of gene segments between 2 non-sister chromatids of homologous chromosomes. And there is known as chiasmata or chiasma. It is the point of contact between 2 non-sister chromatids belonging to homologous chromosomes. And at a given chiasma, there's an exchange of genetic material that can occur between both chromatids. And this stage is particularly crucial as a critical genetic phenomenon called crossing over takes place in this stage. The crossing over involves the redistribution and mutual exchange of hereditary material between 2 homologous chromosomes. Then next we have diplotin. There is a bivalent chromosomes and both on themselves, resulting in separations in the 2 homologous chromosomes. And in the reakinesis, homologous chromosomes completely separate from each other and regain their individual identity. Metaphase 1. It consists of spindle fiber attachment to chromosomes and chromosomal alignment at the equator. During this stage, the spindle fibers attach with the centromeres of the homologous chromosomes, which are directed towards the opposite pole. And we have here the tetrads, which line up at the equator. And when we say tetrads, those are a group of 2 homologous chromosomes that come together to undergo the crossing over event in meiosis. At anaphase 1, homologous chromosomes are separated from each other, and due to the shortening of chromosomal fibers or microtubules, each homologous chromosome with its 2 chromatids and undivided centromere move towards the opposite poles of the cell, because during the chiasma formation, one of the chromatids has changed its counterpart. As we previously discussed about crossing over during prophase of meiosis 1, the double chromatid homologous pairs of chromosomes cross over with each other and often exchange chromosome segments. As the illustration shown in our slide, this recombination creates genetic diversity by allowing genes from each parent to intermix, resulting in chromosomes with a different genetic complement. So during the first cell divisions, there are separations of replicated homologous pair chromosomes, which produces the 4 haploid daughter cells. So the new combination of genes on a chromosomes can lead to a new trace in their offspring. So the crossing over occurs between prophase 1 and metaphase 1. It can also happen during mitotic division. Crossing over is essential for the normal segregations of chromosomes during meiosis. So when the chromosomes go on to meiosis 2 and separate, some of the daughter cells receive daughter chromosomes with recombined alleles. When you see alleles, it is a form of a genes and it is inherited from each parent. So due to this genetic recombination, the offspring have a different set of alleles and genes than their parents do. Alright class, take a look at the diagram. We have a pair of homologous chromosomes. They are aligned where there is a crossing over happens. If you still remember the word chiasma, it is the point of contact between two non-sister chromatids, where there is a swapping or exchange of genetic material that occurs between both chromatids. In the diagram, the genes of capital letter C, small letter c are crossed over with each other, making the resulting recombinants after meiosis. So we have the capital letters A, B, C, the capital letters A, B, small letter c, the small letters A, B, capital letter C, and the small letters A, B, C, which is the resulting recombinant alleles after meiosis. Another stage in meiosis 1 is telophase 1. It refers to the movement of a haploid set of chromosomes at each pole. The nucleolus reappears and thus two daughter nuclei are formed. And then next we have the cytokinesis 1. In animals, it occurs by the constrictions of the cell membrane, while in plants, it occurs through the formation of the cell plate, resulting in the creations of two daughter cells. To recall class, meiosis 1, the chromatids do not separate, which means each daughter cell receives only one copy of each chromosome, the haploid number, and each copy contains two chromatids. In meiosis 2, the chromatids separate and are distributed to each resulting gamete. During prophase 2, each centriole divides, resulting in the two pairs of centrioles. The centrioles move towards the opposite poles, and the nuclear membrane and the nucleolus disappears. While in metaphase 2, during this stage, the chromosomes get arranged on the equator of the cell through the spindle fibers. The centromere divides and thus each chromosome produces two daughter chromosomes. In anaphase 2, the daughter chromosomes move towards the opposite poles due to the shortening of chromosomal microtubules and the stretching of interzonal microtubules of the spindle. In telophase 2, the chromatids migrate to the opposite poles and now known as chromosomes. In cytokinesis 2, the process of cytokinesis is identical to cytokinesis 1, resulting in the division of cytoplasm for each of the four daughter cells formed. And we have here the summary of cell division, meiosis 1 and meiosis 2. What is the importance or significance and application of meiosis? Actually, mitosis and meiosis are both important processes within the body, and as they are the main processes for the makeup of cells. As both processes involve cells dividing to make new cells, it makes it vital for the existence of things that reproduce sexually. The process of meiosis is essential for all sexually reproducing organisms. It maintains a constant number of chromosomes in sexually reproducing organisms through the formation of gametes. By crossing over, the meiosis results in the exchange of the genes and thus causes the genetic variation among the species.
Errors during meiosis can alter the number of chromosomes in cells and lead to genetic disorders. DNA evidence shows that all humans are more than 99% genetically identical. The remaining 1% variance in genetic makeup is due in part to mutations that occur occasionally during cell divisions. So let's take a closer look at how this happens.
As we previously discussed about chromosomes class, they are pulled apart by tiny molecular machines called spindles. And these spindles are made up largely of microtubules and are organized by the centrosomes. Sometimes the spindle does not do its job properly. That means new cells end up with too few or too many chromosomes, and this is known as non-disjunctions. And this means that when that baby is born, it will have more or less than 46 chromosomes. This person will also have either too many or too few genes, and this is called as aneuploidy. Without 46 chromosomes class, the human body will make the wrong number of proteins, and proteins play many roles, like they build and repair tissues like hair and nails. They also create and regulate hormones, and without the right number of proteins, the processes in your body can't work properly. What happens if you have too many chromosomes? If one of the original cells had an extra chromosome, the person will have trisomy, and this is one of the type of aneuploidy. People with trisomies have 3 copies of a particular chromosome instead of 2. And when we say trisomy, there's a trisomy 21, which is very common aneuploidy that involves an extra chromosome 21, and it's also known as Down syndrome. Children with trisomy 21 may experience delays when learning to crawl, walk, and speak. As they get older, they may have trouble with listening and understanding. And there are other examples of trisomy 13 or trisomy 18. They can both cause serious brain, heart, and spinal cord defects. Many babies born with these syndromes only live a few days. What problems do extra X and Y chromosomes cause? So problems encountered having extra X chromosome in a female fetus. An extra X chromosome causes triple X syndrome. It is associated with learning disabilities and organ abnormalities. Some of the symptoms and signs of triple X syndrome, we have delayed motor development, low IQ, delayed speech, abdominal pain, and as well as brain disorder that affects how you pay attention and control your behavior. It is known as ADHD or Attention Deficit Hyperactivity Disorder. Another condition having an extra X chromosome happens in a male fetus. Klinefelter syndrome is a condition that occurs in men as a result of an extra X chromosome. The most common symptom is infertility. Individuals with Klinefelter syndrome tends to have anxiety, depression, impaired social skills, and other behavioral problems. And other signs like a taller, less muscular body than males their age, broader hips and longer legs, larger breasts, weaker bones, a lower energy level, smaller penis and testicles, delay in puberty or go in a person out, less facial and body hair following puberty. Another condition is having an extra Y chromosomes, wherein males with XYY syndrome have 47 chromosomes because of the extra Y chromosomes. And this is known as Jacob syndrome. It is a rare genetic abnormality in which a male receives an extra Y chromosome from his father. The symptoms include language difficulties, problems with sitting and walking, and behavioral emotional issues. People with an extra Y chromosome may also have mild autism and weak muscle tone known as hypotonia. The human body is a complex machine class, and sometimes it makes mistakes. By learning more about human genetics, we can better understand genetic disorders. This will help us find ways to help people who have them. And scientists are now looking for ways to edit DNA so they can reduce the effects of genetic disorders. And this technique is called genome editing. You can make a difference in your community by volunteering to help people with disabilities. To generalize the lesson, we have here the video about meiosis.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis, not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn-out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. And if you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA, since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes, because the sister chromatids are still attached, and we're actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is pro, this actually means before. And it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They're actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line. They're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful of having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes and the ending cells only had 23. So they are not identical to the original and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction, when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study.
Activity sheet number 1 for group activity. Identifying processes. From the given worksheet, you're going to draw and name the different stages of meiosis from the following statements provided from numbers 1 to 9. For your activity sheet number 2, guide questions. How are mitosis and meiosis similar and different? How does meiosis occur in humans? How does meiosis affect the physical characteristic of an individual? Why is meiosis essential in sexually reproducing organisms? And how significant is meiosis in plants and animals? Thank you and God bless.